But what if I told you that everything we thought we knew about nutrient uptake was only half the story?
Plant roots are actually about more than simply extracting nutrients out of soils. They actually are microbe processing structures.
Let us get into the science of the rhizophagy revolution. Welcome to the show, Dr. White.
Thank you, Brandon. I'm happy to be here.
Yes, thank you. Thank you for taking the time today with us and with our audience. We're excited to jump into this, idea that, roots aren't just straws, right? And it's been the way that many people have thought about it for decades, for many decades, that you just put this nutrition into the soil and the roots suck it all up and plants are happy and things grow. But the reality of the matter is things are dying. And so at the end of the day, the reality of the matter is the soil's dying, the plants aren't healthy, but people keep dumping NPK in, right? More and more thinking that these plant roots are just straws. But that's not true, right? They're more like hunters or farmers, I would say. That's what I would say they are,
That's right. That's what we're seeing, Brandon, is that plant roots are actually about more than simply extracting nutrients out of soils. They actually are microbe processing structures, and even more so than leaves. Roots, the way they work is they will attract microbes to the roots. That what happens is that around the meristems, around the growing root tips,
yep.
They'll secrete sugars and essentially exudates, organic acids, and some other nutrients. And those are attractants for soil microbes. So they bring soil microbes to the root tips, and then they absorb some of those microbes into the root cells.
Okay.
And then they use those once they put them inside the cell. They'll take the cell walls off of the microbes, mostly bacteria, but also fungi and sometimes some algae. And so they take the cell walls off, and they'll get some nutrients out of the microbes from those cell walls, and then they'll multiply those bacteria and those microbes inside the root cells. They'll multiply them, and then they'll use the microbes in the root hairs. The microbes will trigger root hair formation, and they'll make those root hairs grow longer and longer. And
wow.
As those root hairs grow, they'll eject microbes back into the soil. Okay, so this is the process that's come to be called the Rhizophagy Cycle, because they take them in at the tip, they eject them from the hairs as the hairs grow, and then they can go back into the soil, acquire more nutrients. And we think it's on the cell walls that they can get some nutrients. And then because they're little, on the cell walls, there are little parts that these nutrients can attach to.
Wow, that's amazing. And that's what's known as the Rhizophagy Cycle.
It goes through the microbes that alternate between the soil environment and the internal environment in the plant. And I should say, I mean, this is something else that your viewers are probably familiar with, the term endophyte, internal phyte, internal plant. Basically, these are microbes that go into plants. So these microbes that I'm talking about, soil microbes, they become endophytes then. And when they're endophytic, in this endophytic stage, this is when the plant can get nutrients from them. So basically, that's it. And the plant, like you said, you mentioned the plants are really farming microbes. And this is what has not been very well appreciated. We've known that there's lots of microbes in the soils, but plants are really managing these microbes closely. It's more like a farming because they're feeding them with nutrients, exudates, and then they're taking them in and they're taking nutrients out of them and they're using them for growth. So it's not just about nutrients, it's about their plant development. If a plant can get the microbes that it needs, then it can grow large root systems, long root hairs, and also branching. And of course, the better the root system, the healthier the plant. So it's also about healthy roots and about healthy plants. They need those microbes in order to grow the robust, healthy root systems. I mean, it actually goes even more complicated than that because I guess I don't want to put it all at once. Maybe.
Well, and we can get deeper into it as we go on through this. But I'll tell you, and that's where that symbiotic relationship really comes in. It's a lot more than we even imagine between the microbes, the plants, and that relationship that they have. And it's pretty interconnected, I would say, and how it actually operates. And so, Dr. White, why does this matter for regenerative agriculture, would you say, in reducing synthetic inputs? Why would the rhizophagy cycle matter?
Well, the rhizophagy cycle is important because that's the way plants do it. This is the way plants have evolved to work, right? This is how they've developed to work. I mean, since plants colonize the land, they've used microbes to get the nutrients they need. And this is the extension of that system. This is the system they use. Plants haven't, plants naturally don't function through simply absorbing dissolved nutrients. Yes, they can absorb dissolved nutrients into roots and root hairs and so forth, but they are actually more active. It's a more active symbiotic process whereby they get their nutrients. And no plant in nature relies on simply absorbed nutrients. They always will have symbiotic microbes associating with the roots and other parts, to the leaves as well. And they're actually seed vectored, these microbes, to go into the seed too. So they're...
Sure. Well, and that's something I was going to say. So even with that being said, you cannot have a sterile growing environment, obviously, correct? Because if you did, you wouldn't have any bacteria, you wouldn't have any fungus or any of those things.
There is no
correct
plant. This is no, there's no even when we try to remove all the microbes from plants, it's difficult to remove all the microbes. We could sterilize them, but there's still some microbes that go through the seed. They're in the embryo, actually, in the seed. So we actually can't remove every single microbe from a plant. There's no such thing as a sterile plant. We like to think we can sterilize them, but we can reduce the community of microbes associated with seeds. That we can do. We can reduce the microbes. But as it turns out, proper development requires of a seedling or a plant requires a community of microbes. So we can reduce the community enough to remove enough of the microbes that we can affect development and reduce development of a plant. And then we see what happens. And basically they don't grow very well like that.
No, absolutely. Absolutely. Especially, and you see it a lot whenever they go in and fumigate land, right? They do it. Some strawberry growers do it. Sometimes there are different types of crops that they utilize fumigants. And you can always tell the next year, right? Weed pressure's high. There's a lot of things that are telltale signs of it, at least that we see. Soil's trying to repair itself. The crops aren't the healthiest. There are issues. So you've already kind of broke this down. Oh, I'm sorry. Go ahead.
They really have a lot of microbes. They're very, because we haven't altered their microbiomes. So, if we mess up a soil, if we try to sterilize the soil, weeds can come in and bring their microbes with them. And that actually, the weeds can help repair a soil too by bringing microbes in, acting as a kind of a cover crop. But of course, they'll outcompete your crop. And some of those microbes from weeds can also inhibit some crops. So it's a tricky situation. But weeds carry their microbes. All their microbes are intact, whereas our crop.
That's very interesting. It's interesting you say that too, because when we were doing some projects with ConocoPhillips, cleaning up land farms, utilizing microbiology, so biological technology, And we were going in and treating land farms. They were on site on the actual well pads themselves. And we were utilizing microbes to basically break down hydrocarbons. It's an easy way to put it. And we would come to some of these land farms that were covered, like you mentioned, with weeds. And we were testing all of these because they were starting to receive fines from the EPA for having these land farms at the well sites. And so we would go and we would test them. And the ones that were just barren, that they had been turning the soil, the hydrocarbon levels were they were off the charts. But we would go to the land farms that had weeds growing on them. And every one of those little land farms that had weeds growing over the top, they were in perfect condition. Like literally the soils were perfect. There was no hydrocarbons found. Nothing was there. So it's interesting you bring that up. It just, it verifies it over and over. Farmers don't want to plant weeds, right? Cover crops are weeds, but controlled weeds.
We actually like to study weeds. And I have one graduate student who's focusing on studying the microbiomes of weeds and how they work. But the reason we want to study them is because they have the microbes intact, right? And they use those microbes. So we could see how the weed is using the microbe. And we could even see, which microbes are there, take some of these microbes off weeds and use them on crops. So we think we can learn a lot more about how the plant actually uses these microbiomes, these microbes, and then take what we learn, apply it to the crop plant. Where we've lost a lot of the native microbes.
Sure. No, that's amazing. And you've kind of already went over this. I'm just going to go over it again real fast. We talked about it a little bit, the Rhizophagy cycle and kind of the breakdown. And you talked about it, the attraction, which are the exudates, correct?
Yeah.
At the root tip, which lure specific bacteria. Then you have the entry, right? Which you went over that. It's give us the brief description of how the microbes enter the root cells. So do the root cells actually open up and actually eat them?
Well, we don't really know. The Australian group, this process was actually first identified by an Australian group, Queensland, Australia. And they published a paper in 2010 in a journal called PLOS One, open access journal. So you could find that. It was the title was Turning the Table, Plants Eat Microbes as a Source of Nutrients. Okay, so they really get credited with discovery. What we did in my lab is we studied the process to try to understand how this is working. We were actually working on how plants get nutrients from microbes even before that, but we didn't think they were inside the cells. But after they published that, we noticed they're actually inside the cells. So what they do and what they proposed, the Australian group, they proposed that these the plants actually will take them in through the process of endocytosis. Okay, so it's like basically eating them. Okay, that may be the case. We don't know for sure. We have some other ideas about it, but basically the answer is we don't know. We can see them going in. So we know they're taking them in. We can see them after they're taken in. We can see what happens to them after they're taken in. Basically, they get their cell walls removed. We could detect reactive oxygen that the plant puts on them to digest off the cell walls. So we know what's happening. But what is the exact process whereby the plant takes them in? What is that mechanism? It's not clear. There's like 3 different hypotheses to explain that. And there's no point in speculating on which it is, but the fact is we'll take them in. And that's a spot. The meristems are where the cells are dividing. So the root tip meristem cells are dividing, right? And they're growing fast. The walls are very thin there. The walls are plastic.
Okay.
Right? They can expand because their plant is growing. Those cells are growing and expanding. So the walls are not hard like they are at other parts of the plant where the walls get hardened, right? They're soft. And so the microbes can go right through.
Interesting. Yeah. And then that's what I was going to say. So the entry is really, it's still kind of up in the air. Like you said, 3 hypotheses that potentially, they don't know. Bottom line is they're getting in the roots.
See them going breaking through. But what is the? I
Yes.
mean, this relates to I mean, this is I mean, we could tell you they're going through, and we can see them going through, or if we knew a lot more, we could go into the detailed mechanism of exactly what's happening till they go through, and then that would be more complex, right? More complex. But,
yeah. And the research is being done, I'm sure, and at some point it'll come out. But the reality is they're inside the roots. They're dump trucks dumping nutrition in there, right? And you did kind of mention that. The stripping process is the next thing, which is really interesting. The reactive oxygen to strip the cell walls off.
Exactly.
Right? Which then that kind of releases the nutrition into the plant, correct? When those cell walls are stripped.
Coming off, and the walls frequently, when these bacteria are in the soil, other nutrients like divalent metals will attach to those walls, little receptor areas, so they'll attach there. And so when those get oxidized, when the walls are taken off inside the cells, mind you, inside the root cells, when they're taken off, those metals are released. And some of those metals, like manganese, are in very high concentration when plants do rhizophagy cycles. So some nutrients are really, really important. Rhizophagy is really, really important for certain nutrients.
Interesting. You had to even enter the plant itself, period.
Yeah.
Wow. That's amazing. And then can you describe kind of how the surviving or the protoplast microbes trigger root hair growth then to exit back into the soil? Is that what you're talking about, where they're coming back out of the root tips back into the soil then, where the new root tips are being actually formed?
I could tell you, yeah. What happens is once they enter into the root cell at the root tip, they're oxidized, they lose their cell walls, and then they become these little protoplasts. They're actually called L forms for bacteria, and we also didn't name that. That's from the 1940s or 50s. They found these in the Lister. L form, L refers to Lister Institute, so they found them in the Lister, discovered them in the Lister Institute. But they're well known. Well known, these protoplast forms. And what happens is those protoplast forms can divide very rapidly. And so the plant, when it begins to form root hairs, and it only forms root hairs because the bacteria are in those root cells. If we remove all the bacteria, there are no root hairs to form. So they're inside those root hairs, and they will, at the tip of the root hair, they'll accumulate at these tips of the developing hairs, and they produce hormones. Hormones, things like ethylene, nitric oxide, and that causes an elongation in the hair. And when the hair elongates, some of those bacteria are ejected out into the soil, because when the hair elongates, the tip of the hair gets little pores in it. So the bacteria go out right out of those little pores in the tip. Then the bacteria that remain in the hair, so there's a little growth spurt. The bacteria remain in the hair, then circulate around through a process we call cyclosis. But what the plant is doing is it is breaking these microbes up. It's really replicating those bacteria, those little bacterial protoplasts, so there's many more copies. Then they accumulate in the tip again of the hair, produce hormone, cause another growth. And then some of those bacteria are ejected out. But some on the side again remain, some remain in the hair, and then those are replicated again until they increase and accumulate in the tip. So you get these growth spurts that keep happening. At every growth spurt, microbes are ejected back out into the soil. When they're ejected back out into the soil, they reform their cell walls and any flagella, whatever they need to survive in the soil, right? The soil capable. So the plant is, keeping them captive in a sense, inside its tissues, inside its cells, and then it releases them and actually will put from the tip of the hair, we'll put a little bit of exudate out so that the microbe can rebuild its cell wall and be soil capable again, to go out and survive the soil.
Yeah.
It's really
wow, that's amazing.
It is, and it basically is a process that is under plant control. It is plants,
Yes.
plants farming microbes, plants farming microbes, plants managing these soil microbes. And
managing cattle, right? All in the soil. It is. And they're telling them what they need. That's what's amazing is the plant's like, okay, hey, I need this nutrition, or hey, I might need this, or hey, I need this now. And that's where those exudates then are produced, and then the proper bacteria are farmed and brought into the plant. And let's talk a little bit about endophytes. I like to say they're the interior design of plant health. Can you kind of share, endophytes that live in the leaf, stems, and seeds of plants? Like, are there specific endophytes that live in there? And I'm not saying tell us all of them. I'm saying some very specific ones that live in the leaf, stems, and seeds of every plant.
Well, they're mostly, these endophytic microbes are mostly of two kinds. One are fungi, and fungi are definitely there. Fungal endophytes tend to be less common than bacterial endophytes. And there are different bacteria that will go into leaves. There are many, put it this way, roots absorb a lot of microbes, right? They will absorb a lot of microbes into the tissues of the root. Okay, and so you have a complex community of microbes inside roots and on surfaces of roots, but also inside the cells of the roots and between the cells and between the tissues in the intercellular spaces. Okay, so you get a lot of microbes in roots. A subset of those microbes are vectored up through xylem and sometimes phloem, and or they ride in the meristem from the seedling, and a subset of those microbes are actually in the leaves and end up being seed transmitted, going to ovules and get on the surfaces of seeds and transmit with seeds. But so it is really, they're not unique from the soil microbiology in that sense. They are just they are the microbes that the plant most critically needs. A subset of those microbes that the plant critically needs for seedling survival. And that's how it actually works. And that's what the mama plant is doing, is moving those microbes up to put on the seeds and to use in the leaves. They do things that it is, this is a whole area. There's a lot of ecology that goes on here too. They will actually colonize. In some cases, they'll colonize chloroplasts. Like we're doing work with some organic corn full of microbes. And one of the things that we see over and over again is that these bacteria that go into the leaves will actually form biofilms or in those leaves around chloroplasts in the cells, right? The chloroplasts are in the cells. So they actually will then, there's an internal symbiosis that happens with the bacteria that involve these chloroplasts in a biofilm. And they will go inside the chloroplast to some extent. So it really is, there's a whole ecology inside plants that's not appreciated, that's not known. And you think, well, we should know, all of the stuff that's going on. But in fact, we don't. And the reason is because, people in different fields, they're disconnected. Say, for example, take the field of physiology, plant physiology. And they don't use microscopes very much. They don't look, they don't know how to track. They just don't. It's not in the mindset. It's more of a physiology is very, very important. But explanations, when you have an explanation for what's happening in a plant, say it may be, increased photosynthesis, right? Increased photosynthesis. So the explanation, if you're a plant physiologist, the explanation is going to involve, some physiological mechanism, right? But it's not going to involve a symbiotic association that might impact the efficiency of chloroplasts, right?
That you can see at the microscopic level.
With the microscope, you could then do experiments. So because those fields are isolated, they would not even think of interpreting what they see as having anything to do with microbes that are inside those plants. And it's just inside the leaves,
absolutely,
or inside the roots or wherever they are. It's just, and that has to do with the structure of science in various fields and the way they progress. I mean, it is
sure. Yeah, no, absolutely.
Part of the issue why our knowledge is so fragmented, and I think that's what I'm trying to say is, and the different fields are so isolated from one another, and their explanations they can't see. It's a kind of a blindness. You can't see any other explanation unless it has to do with all the stuff that's well known.
Yes.
In different fields, so.
And then you have to go out and find that information, right? Like it's like say the research you're doing. Now you have to have a plant physiologist want to go out and now research what you're doing. And,
what I'm doing comes from a different field. What I'm doing comes from this burgeoning, this increasing, growing field, what I call endophytology, right? People study, basically people worldwide studying microbes inside plants and how they work, and that's a separate field, but a growing field. People in plant physiology or whatever if it's genomics, metagenomics, or whatever they're looking at, they wouldn't look at it that way. They wouldn't, if they're looking at the way gene expression occurs, you have to know there's a microbe there, and you have to ask a question specifically about that microbe, and then you have to have, and these are little tiny things, then you have to be able to measure little things about that microbe in there, and the big picture thing, you just doesn't work, and
sure.
so there's this isolation, and that makes people blind. They really are blind to these, this ecology inside of a plant.
Yeah, no, seriously. And, you've mentioned a couple of times now about seed-borne microbes, right? Like there are seeds that carry microbes with them, right? And I have, I guess, a couple of questions here. For one, can microbes repair genetic drift in a plant? Like if you have a plant that you're taking cuttings off of or something like that, can that happen? How does the plant take that's microbiome and put it to the seed? Does it just have specific microbes that it puts in each seed? Like how does that work?
So yeah, that's a good question. Okay, a number of different questions that you asked there. So I'll go with the last one. Okay, so how does the plant vector microbes to the next generation, right?
Yes.
There are structures on seeds that actually carry the microbe on them, carry microbes on them. Like, for example, if you consider a grass seed, a ryegrass or tall fescue, something like that, you got a seed coat, and closely adhering to that is organic material called lemmas, little leafy material, right? Dry leafy material that carries microbes, on it.
Okay.
So they go on it. Other seeds, take for example tomato seed, will have microbes on the surface, but oftentimes our fermentation process will alter those microbes on the surface. But they also have microbes internal. If you cut some of those seeds open, you can see they have biofilms inside them. Okay, so
inside the actual,
them, right? So there some critical microbes are actually vectored internally. If you consider something like a tomato, the natural way that tomato seeds are vectored, they're not. What people do is we'll take those tomato and we'll ferment it and we'll get all the organic material off so it gets degraded, and then we'll take the seeds that are pretty clean and then dry those, right? Wash them and dry them,
Sure.
and there we have, right? But in nature, that's not how it works. In nature, a tomato is actually a berry, right? It's a kind of a berry, and it's a berry that's consumed by mostly birds or other animals, but birds especially, and they will consume the berry, and then it goes to the gut of the animal. And if you consider the gut, it's full of microbes, and so it goes out into the feces, right? It goes into the feces of the animal, and in doing so, it picks up a lot of new microbes. So we look at the microbes associated with tomato. A lot of those microbes are actually gut microbes. They're the same ones that we see, the same micrococcus, acinetobacter, E. coli. They're actually animal. They're the same ones you see on animals, which is different than, some, for example, grasses. Gives different endophytes that are there. So the ecology has something to do with how those, the particular ecology of the plant, what microbes are there and how they pick up microbes. So the natural process for something like a tomato would be going through the gut of an animal, right? And then that's the seed vectoring process. I mean, the wild tomato, right? Our cultivated is totally different, but not so
100% different.
Honestly, it's not so much different. If you look at when I was a young guy, I did young man. I doing work in summers. I would cut grass, and one of the places we had to cut grass is around the sewage plant, right? And so around the plants, you see plants around the water settling plant for cleaning the sewage, right? You see plants growing, and frequently the plants you see growing there are tomato plants, and that's because they go through the guts of humans, and they survive.
Interesting.
That's their niche. But yeah, it's anyways.
Man, I tell you that that's interesting. Yeah. Now that we know that, let's look at the guess and stress connection now. Would you say that modern chemical or conventional farming, high usages of fungicides, high salts, might be blinding the plant by killing these internal partners before they can even start their cycle?
Yeah. I think that the way we grow plants, crops, does not foster the plants acquiring microbes that help them to acquire nutrients, certainly, and to increase stress tolerance in the plant, for stress tolerance. So, yeah, I think it does. Now, we don't know all the effects. I mean, one effect, for example, is if you're putting nitrogen on the plant, nitrogen fertilizer, that reduces the need for the plant to acquire any nitrogen fixers, right? Either in the soil around the plant or absorbing it into the plant. And that's what happens. It will turn off. Putting nitrogen there will turn off nitrogen fixation genes and those microbes that are absorbed. I mean, some microbes will be absorbed, but not as many, and certainly not the diversity that you do when you're not putting all the fertilizer on. It definitely is a negative impact. It's there was another, and the other issue is stress tolerance. I mean, I mentioned that. The stress tolerance effect is significant with microbes in the plant, and you need to have a diversity of microbes or a particular microbe that's stress tolerant, that gives this stress tolerance, so not all microbes will do it. And so the more that the soil microbiome is damaged by agrochemicals, the less able these crop plants are to get beneficial microbes from those soils.
Almost kind of turning them into addicts per se.
It is addictive. I mean, it is the more you use chemistry, the more you have to use chemistry. I mean, anyone that knows that it grows crops or grows turf, right, knows that once you start down this road of chemistry, you have to use chemistry. I'll give you a concrete example with a turf grass, with Bermuda grass, right? Bermuda grass.
Okay.
So
yeah.
The way Bermuda grass is cultivated frequently, when we have Bermuda grass seed, the seed has a hull on it, and that hull seed reduces the evenness of germination because it has to, people don't like that. Okay, so they knock that off. And when they knock that off, that hull, what they've done is they've removed the structure that carries the microbes on the little Bermuda grass seed. And then the seed becomes now vulnerable. It germinates fast, but it's really vulnerable to pathogens and everything else. So once they do that, then they're going to have disease if they don't then coat that with fungicides and, it's other chemicals. But of course, that's because they have to coat it with the fungicides because the bacteria are gone that would not only provide nutrients, but would protect the seedling from pathogens, from damping off. So
absolutely.
You then become dependent on that. And in soils, if you look at a healthy soil with rich microbial microbes, the plants are going to be they're going to grow well. They're going to use microbes to grow, and disease will be at a lower level. But once you start fertilizing, then you're going to see what happens is an increase in disease, and then you have to use fungicides.
Yep. Absolutely. No, absolutely.
That's an addiction.
Go ahead. Yeah. So, and that's what I was going to say. And that's something that I look at as well as like the simple one is the nitrogen, right? If you put all the nitrogen fixing bacteria, on their lazy chairs, they're not wanting to do anything for the soil if you're providing all the nitrogen. The atmosphere is nitrogen. Use it to your advantage, right? So we go through this a lot, and these are the first steps that we have to take a lot of times with producers or ranchers or people that have been on the cycle of conventional inputs. It's like, look, if you're going to put inputs in, make it an investment that's going to pay you back. Don't just make it an open-ended pipe at the end that you just keep putting money in and it just keeps flying out the other side. Don't do it. If you can, and there's time that you have to take to convert them away from it and educate them and all those things. It takes time, but more and more people are starting to see it. So let's talk a little bit now about the practical implications for the field. So we talked a little bit about this now, the fertilizer efficiency. So if plants can farm microbes, for nitrogen and minerals, which we've been talking about this whole time, how much do you think we can reduce NPK without sacrificing yield? Say on a conventional, and I'm not saying one crop, corn, I'm saying overall, just a percentage. What would you say would be safe to be able to start pulling back fertilizers?
I have no basis for answering that, Brandon. It would be right off the top of my head. And I could not. I mean, I could tell you this. What I've heard is that it's a couple of people mentioned numbers, people who know with more experience than I have at that, more practical things than I have mentioned a 30% decrease, right? A 30% decrease in fertilizer or more. Some people have gone all the way down, to very little, to only 30% fertilizer, what they were doing, but mostly I've heard that you could get about a 30% reduction in fertilizer easily with no loss in yield. And I've heard if you knock it all the way down, some people say, well, there's immediately a 10% loss in yield, something like that. But then after a few years, you can build it up. So it's something that this is the only thing I can say, and I'm sure that's going to be crop dependent, and it sure. Really it's going to depend on the soil. One really has to build up soil and build up the microbial community in the soil in order to build up soil organic material. Use cover crops to try to increase the microbial activity in soils, and sure. As that happens, then there will be less of a reduction. I mean, you could go further and further down.
Let us talk about your findings that certain microbes help crops but inhibit weeds. Can we use biology as a selective herbicide?
You can. It's absolutely correct that certain microbes will benefit certain specific plants and then inhibit other plants. Okay, it's kind of like a biological allelopathy effect. Okay, we even put a patent out on that. The problem is that in soils that are highly diverse, it's hard to get these inhibitory microbes to go into the target plant that you want. They have their own microbes, gotcha. And they can essentially use those microbes to push out these. At least that's what we think is going on, to push out these other microbes. What happens?
There's kind of a little war going on, huh? You got these guys, they're setting up their cavalry over here, right? All their turrets.
Plants are in control, right? And weeds in particular have a lot of microbes in them, so they have their own microbes. So it's tricky to get them to take these other microbes in. And these other microbes, the way they work actually is they are oxidatively resistant. So they when they go in, when they get taken in, these weeds, the weeds aren't able to control them. They using reactive oxygen, and so those microbes will then go out of control and essentially take over the machinery of the weed. So things like that are easy to do in the laboratory, right? Mm-hmm. But hard to do in the field. This is the story. This is the story of agricultural science innovation.
Hey, but you know what?
It's harder in the field. Yeah. What was it?
But I think you answered the age-old question. That's why weeds grow out of cracks, because they have so many microbes.
Yeah, they are.
Right? In concrete, right? It doesn't matter. They'll grow anywhere,
Yeah.
They will. Yeah,
and they're also resistant. Oftentimes they're, like I said, I have a student working on weeds, and we've been looking at Poa annua and the microbes that are vectored there, and it's really interesting. Those microbes will actually are giving the seedlings resistance to herbicides. It's a certain level. You can overwhelm it. If you put enough herbicide there, you overwhelm the resistance. Sure. But the seed microbiome on Poa annua gives herbicide resistance, and we get heavy metal tolerance, so stress tolerance. It's all kinds of stress, including herbicide stress. It's
That's
really
amazing.
probably a lot of wild plants are doing the same thing, but some of this we've lost from our crops.
Absolutely.
Yeah.
Absolutely. And that's what that's what I was even going to say now with some of these microbes that you guys are finding, some of the research you're doing, that's that stress tolerance coming in, right? Because like you said, how do microbes provide defensive mutualism, protecting plants from heat, salt, and pathogens? And so there are specific microbes out there that will do that, or that can even operate in, say, salty soils or high pH soils. I mean, I think we've just scratched the surface. Well, I know we have. We don't really even know everything that's out there. And then let's talk a little bit about the role of soil testing. Explain how understanding microbial biomass relates to the success of this cycle. So by knowing your microbial biomass of the soil, how does it, basically give you this success to the cycle?
Well, it reflects the microbial biomass reflects the numbers of microbes that are out there. It doesn't necessarily reflect diversity of microbes. So you want more than just the biomass, but you want to know that there's good organic material there in the soils for accumulating these microbes, so the microbes can survive. But the biomass is important because, assuming you have a good diverse soil, the biomass tells you there's enough bacteria there for these plants to get hold of, to absorb into the roots. So that is an important measure, and there's a direct effect between the rich microbially rich soils, and healthy antioxidant and health compound in the crop, right? Health compounds in the crop, like phenolics.
Sure.
Right? There's a direct connection. The more you have a diverse microbiology in the soil, good biological soil environment, the healthier are those crops going to be. The more of the health compounds like the phenolics and other antioxidants are going to be in those plants. And not only that, the more the plants are going to carry the microbes from the soil, because those microbes go into the plants. And if we eat those plants, those crops, particularly if they're uncooked in salads, for example, that'll go right into our guts. So there's a direct connection between soil health and human gut health.